Hi, my name is Katelyn Overstreet, and today I'm going to provide an overview of the research that I'm currently doing regarding leaf litter and overall black mangrove growth. Mangrove trees are among some of the most highly productive coastal ecosystems. Because they're located at the land sea interface, they not only protect the structural integrity of the coast from storms, floods, and erosion, but function as nurseries, breeding sites, and microhabitats for a diverse range of terrestrial and marine organisms. Consequences of global climate change, such as rising sea levels, increases in temperature, and ocean acidification, are posing significant risks to mangrove ecosystems. There's a global loss rate of mangrove ecosystems estimated around 1 to 2% annually. This phenomenon could potentially threaten species biodiversity. Leaf litter, which falls from trees and other forms of vegetation, supplies the environment with energy and substrate upon its decomposition, and thus it functions to shape the overall chemical environment. Amidst global climate change, understanding the nutritional value of different litter types is of great significance to the overall health of coastal ecosystems. The aim of this research is to measure mangrove health and thus nutritional value derived from three distinct categories of leaf litter: frozen leaves, senescent, and green leaves. 38 trees were planted in a sand medium lacking any nutrition with a water source of only DiH2O. Approximately 2 grams of leaf litter of each treatment type, senescent, green, and frozen, were added to respective experimental groups and equivalently to a control group in the form of paper scraps. Each week, 3 sets of measurements were taken for each experimental group: internode height, overall stem height measured from the base of the pot to the top of the tree, and leaf count. So here are the results for mean growth of each experimental group. As you can see, senescent showed the highest growth rate in terms of overall height, followed by frozen, green, and lastly control. This set of data was a little bit surprising. I didn't end up having a standard error that was a little bit off. And as you can see, both green and control actually had negative growth rates, which was interesting, which I contribute to a few troubleshooting errors, which I will explain in the next slide. Over the course of this experiment, we've had to do a decent amount of troubleshooting. And I think the biggest problem that we've run into overall has just been sand loss, which in turn affected in my opinion, the weekly measurements of the overall growth. The pots that the mangroves are actually planted in have a pretty decent sized hole at their bottom, and this is just so that the roots can extend down and absorb and collect the nutrients provided by the litter treatments themselves. But as you can imagine, shifting the plants and moving them results in a significant amount of sand being lost. In my opinion, I believe the biggest source of sand being lost was actually a few of the issues we ran into in terms of having to repot the mangrove trees. So initially we had them in Tupperware-like containers, but we did not have very much room on the shelves. If anyone's ever seen the plant lab, it's a pretty small space. And so to overcome this, we decided to put them in Ziploc bags individually. And of course, the structural integrity of those did not last long, and they soon acquired holes and broke. And so at this point, we had to come up with another solution. And we ended up going ahead and putting the plants of each group into single planters. That way, we created a lot more space and didn't have to risk any tears or water leaks or anything of that sort. Taking into consideration that the mangroves did lose quite a bit of sand and that possibly was affecting their overall height measurements, I went ahead and created a separate graph, a hypothetical adjusted mean growth, And this graph was constructed basically just using the first stable point of my measurements. So when there wasn't huge decreases in overall height measurements between the weeks. So the first constant point of measurements. And as you can see, that did go ahead and shift things a little bit. I had a way less standard error than the previous measurements. And in this graph, you can see that frozen leaves, as opposed to senescent, actually showed the highest growth rate, with green coming in third place, and lastly, the control. And these results actually align much better with what would be anticipated with each experimental group. So here are the results for overall internode elongation. So far, frozen has shown to have the highest rate of elongation, followed by green. And interestingly enough, senescent and control have ended up having equal rates of internode elongation. These results are a little bit surprising just because, as shown in previous slides, senescent by far, even on the adjusted mean growth graph, have shown significantly higher overall growth rates. So it was a little bit interesting to see that here green had substantially higher rates of internode elongation. So here are the results of overall net leaf loss for each of the groups. This was measured by essentially just doing a total leaf count on a weekly basis for each of the groups. And as you can see, green actually has shown the highest amount of leaf shed, followed by senescent, frozen, and lastly control. I think these measurements might be a little bit off just because initially during the experiment we did have to troubleshoot. We were shifting the plants around a lot of breakage occurred just between different trees during rearranging them along the shelves. And so I think these measurements may take a little bit more time to show any substantial difference between each of the groups. So overall, our results definitely point to frozen litter treatments as being the most nutritionally valuable of the groups. Green and senescent treatments appear to provide greater nutritional value, which would be expected than that of the control treatments. However, green and senescent treatments have shown quite a bit of variation in terms of measurements. Senescent leaves show increased overall growth rates and decreased net loss of leaves compared to green, while green leaves show greater internode elongation. Given that it's still pretty early on in terms of the experiment, I anticipate that over the weeks to come, with a little bit more data collected, we'll see a significant difference in terms of distinguishability between the three groups. So I'm looking forward to sharing the final results.